Ram. So I'm working with Sua from Australia. So first of all, I would like to thank for this wonderful planning of this sort of retreat. I would like to thank Sua and TJ and this committee to make this happen. So far, I would like to share some of my knowledge recently, what my curiosity I acquired about cancer genomics. Probably this can help everybody to understand about how the genomics in the cancer cells. So why we want to study cancer is most of the people already studying about the cancer related genes, how what are the functions of the genes. So So here, globally, and I got to data for 2007, there are 12 million new cases that were diagnosed, there are 7.6 million cancer deaths happen. And most of the deaths, 70% of the deaths happen in poor countries, developing countries or low income countries, because either they don't have complete knowledge of this picture or not proper diagnostic techniques. If this continues like this, by 2050, there will be more cases, you can see diagnosed cases, and then there is 75 million people might suffer due to not having enough knowledge of this cancer genome. So in the United States, also there is a lot of the disease is severe, and most of the people get, due to the hormonal effects, they get either breast cancer or prostate is the majority fraction, and most people get lung cancer also. But most of the deaths happen due to the lung cancer. And there are other cancers also people do get, then that's a little bit less compared to these cancers, major cancers. So then a lot of people, they call cancer is a genetic disease. If you understand the genome of the cancer cells, probably you can able to understand most of the picture how these cancer cells work. So in that, majorly they categorize two kind of mutations. One is, some mutations comes from heredity, and some other just somatic mutations which you can acquire over your lifetime because you are exposed to different kind of environments. So people, when they are predisposed to heredity, when they get some abnormal gene through heredity, when they are exposed to some of this environment, they are more highly likely to get cancers than others. And mostly the tobacco is the killer of a lot of people. Then other radiation and chemical pollution, especially in developing countries, they are facing nowadays this problem. So as we know knowledge, like a lot of people studying about how tumor suppressor involved in these oncogenes, inhibitory and telomerase, our lab is studying on these genes. But there are more genes are involved in this process. Probably by sequencing all the genome, probably can able to add more genes to this these genes. Then we will get more opportunity as fundamental level to study about all these genes involved in this process. So the intuition came to do the cancer genome sequencing from Human Genome Project. Because of Human Genome Project, there are sequencing techniques, second generation sequencing techniques, they improved a lot, and now you can get whole genome for $1,000. So once you do the mapping of human genome, now it's a time to go and look at the cancer genome using similar kind of methods. So the goals of cancer genomics is that to obtain spectra of all somatic mutations in human tumors, and to characterize all these tumors, and then create an extensive database of what are the driving mutations and passenger mutations involved in this process. And once you get, then you put in a data portal. There's a lot of the institutes they are doing right now, and you can get from them. Then pharmaceutical companies, they start doing developing drugs according to what are the mutations involved in these tumors. So then, recent 2008, there's an international cancer genome consortium formed, so that people cannot duplicate the same work, so that they can the cancer genome consortium, they organize the things so that some people do some kind of cancer tumors work, and some other people do some kind of. So each cancer they are investing $20 million to sequence that cancer. Then they put the data will be accessible to the public later. So mainly in the just say the cancer genome atlas, you can go to that website, you can get a lot of the data which they got it from cancer genomics. And the UK Sanger Institute is doing this job. So how you sequence? So how you do this sequence analysis? They take the genome from normal cells of same patient, the cancer patient, and they do take the genome from cancer cells. And once they get data from this, then they compare with the genome and they subtract the mutations and all of the stuff from that. As you see that cancer genome is pretty messy compared to human genome, you see a lot of extra copies and some of the deletions you can see. It's not an easy job to do that people are working on to get this as soon as possible, this data out. So another thing, they are going ahead and to see that cancer develops over the time, and most of the cancer sometimes takes several years. And to get the data from normal sense and initiated and mild, and if you can get data from all over the range, then what you can do is that you know that what are the mutations. First started with some kind of passenger mutations, then there are driver mutations most of the time they appear somewhere here, and you can able to see that what are the pathways involved in that in this way. So next slide. There's something up.
Yeah.
Sorry, it is a crash. Hang on a second.
Jeannie, this is your computer.
That's mine. It's still like the program crashed.
Maybe we should get another.
Yeah, no, but I can't. This thing is crashed.
The tissue level can give the answer of lot, like giving probably that can give the information of origin of this cancer. So how this cancer progresses, where we can also develop a lot of therapeutic benefits. So this is what the aim is, that each cell, as you progress and you see that over the time you develop a lot of mutations, and most of the time earlier stage you develop only the passenger mutations, and over the time you go, there's a star which are called driver mutations, and which is generally they drive the cancer cells into the progress into the cancer type. So the more and more driver mutations occur over the time, then they're going into the cancers. So probably whole genome mapping of all the system can give us the very clean picture of how we can understand this cancer and what genes we can target using the drugs, and how we can, even this can be useful for fundamental research also. So, I mean, like this, there are several papers recently appeared, because I'm not going into that feature, because in Nature there are 2% of papers contributed due to cancer genomics. Actually, that's what it makes very interesting. And the work which they say that study reveals genomic similarity between the breast cancer and ovarian cancers. Probably this can help a lot of pharmaceutical companies. They develop the drug treatment maybe which can be similar to the both cancers. And also a lot of driver mutations recently found, which also previously not known. So, what are the output from cancer genomics? Probably, as this guy said, that if you get all the sequence from this cancer genomics, probably you can able to say that mutations happen in certain kind of flexible regions of the DNA. So probably you can acquire all the data from these portals and to check out that using bioinformatics to how that happens. So what they found is that each cancer is different, each patient, and that they have found a lot of heterogeneity across these tumor types, and the high rate of abnormalities also they found means there are a lot of driver mutations and passenger. So far, also people used to believe that there are 5-6 driver mutations because when they mutated these proteins in the mice or other system, generally there are 6 proteins when they delete or knock out, generally they tend to develop cancer. But they say now, people predict that there are more like these driver mutations that involved in this cancer. And 1.6% of genes mutated out of 2,100 human genes, which also output of this cancer genomics, and also some cancers they need a lot of driver mutations, some other cancers they don't need too many driver mutations to send to get cancer. So this all information is very, very useful for future development therapeutic techniques. And but future I would tell you that along with this the sequence of if you sequence the epigenomics, if you know about these cancer cells, epigenomics and transcriptomics, if you combine all this data, that can give a clear picture of how one gets cancer and how you can cure the cancer.